Welcome to the deep dive. Today, we're really getting into it, right down to the microscopic level. We're talking about energy.
Yeah, not just feeling energetic, but the actual, chemical and biological nuts and bolts that keep everything running 24/7.
Exactly. And it's something we all experience, right? This weird energy paradox.
Oh, definitely. You could be listening right now feeling totally wiped out, just drained, heavy, can barely find the motivation.
And then, maybe an hour from now, something clicks, an interesting idea, an urgent task, and boom, you're suddenly alert, focused, good to go.
That up and down feeling, that energy roller coaster, that's what feels real to us.
But here's the thing we're digging into today. The actual machinery inside your cells making that energy, it's pretty much running constantly. It's tightly regulated. It doesn't just flip on and off like a light switch.
Right. So that's the core question, isn't it? If the molecular engine is always chugging along, why do we feel so wildly different from one hour to the next?
And that's our mission, to unpack this. We'll look at the chemistry, the pathways, and show you that your subjective feeling of energy, it's not just about how much fuel is in the tank.
No, it's more complex. It's what we call an emergent property. It arises from this whole layered system, hormones, brain chemistry, and crucially, whether fuel is getting to the right place at the right time, especially the brain.
Okay, so let's start at the absolute foundation, the molecule that pays for basically everything in life, adenosine triphosphate, ATP.
Right. And to really get ATP, you have to understand where most of it gets made, efficiently anyway, and that's the mitochondrion.
Ah, the classic powerhouse of the cell. We all learned that in school.
Yeah, and it's true, but honestly, that nickname sells it way short. It's so much more than just a power plant.
It's more like a central hub doing a lot of different jobs.
Precisely. Yeah, besides churning out energy, it's deeply involved in cell signaling, guiding how cells differentiate and grow. And this is critical. It holds the keys for programmed cell death apoptosis.
So if a cell gets the signal it needs to self-destruct,
The mitochondrion can release specific molecules, like cytochrome C, out into the main cell body, the cytoplasm. And that kicks off the whole self-destruction sequence. It's a major control point.
which means its physical structure, its architecture, isn't just random. It's designed specifically for these jobs, power and regulation.
Absolutely. The design is the function in many ways. You've got this double membrane system. It creates these 5 distinct compartments. The outer membrane on the very outside.
Okay.
Then the space between the membranes, the intermembrane space. Then the inner membrane, which is highly folded.
Those folds are the cristae.
Right, exactly, the cristae. And inside those folds, you have the very core, the matrix. Each space has its role.
And if the matrix is where some key reactions happen, like the Krebs cycle we'll get to, those folds, the cristae, they must be super important for the energy production part.
Oh, they're the genius part of the design. That inner membrane is really selective, like a strict border guard. But folding it up into cristae, that massively increases the surface area.
More surface means more space for the machinery.
Precisely. It allows the cell to pack in literally thousands of the protein complexes needed for making ATP. We're talking about the electron transport chain, the ETC, and those ATP synthase enzymes. Without that huge surface area boost from the cristae, the energy output would just plummet. It wouldn't be enough.
And you can actually see this difference in real cells, can't you? Cells with higher energy needs have different mitochondrial architecture.
You can? Take heart muscle cells. They never stop working right. Their energy demand is enormous. So mitochondria can take up something like 40% of the cell volume in heart muscle.
Wow, almost half the cell.
And even more telling, those heart mitochondria have about 3 times the number of cristae folds compared to, say, mitochondria in liver cells. Liver cells are busy, sure, but their moment-to-moment energy demand isn't as intense.
So the cell literally builds the infrastructure it needs for the job.
Exactly. The internal architecture is tailored to the tissue's energy budget.
Which means if this carefully built structure starts to break down, if the mitochondria become dysfunctional, the consequences are huge and probably hit those high-demand tissues first.
That's the crucial point. Mitochondrial dysfunction is now linked to a really wide range of human diseases, things like type 2 diabetes, but also neurodegenerative conditions like Alzheimer's, Parkinson's.
Really fundamental stuff.
Yeah. And often the root problem comes down to either not making enough energy or making too many damaging byproducts like oxidative stress during that energy production. It just highlights how vital this one little organelle is.
Okay, so mitochondria make the stuff, but ATP itself is the actual energy currency. What is it, chemically speaking?
So ATP, adenosine triphosphate, It's built from three parts. An adenine base that's one of the building blocks of DNA, too, a ribose sugar, and then the key part, a chain of three phosphate groups tacked onto the sugar.
And the energy isn't just floating around in the molecule, it's stored specifically in the bonds between those phosphates.
That's right. Particularly in the bonds linking the second and third phosphate groups. You can think of those as high-energy phosphodiester bonds. They're like tightly coiled springs, just waiting to release their energy.
So when the cell needs to do something, contract a muscle, send a nerve signal,
It snaps that spring. It breaks off the last phosphate group through a reaction with water that's hydrolysis. So ATP becomes ADP, adenosine diphosphate, plus a loose phosphate group. And that breakage releases a neat, usable packet of energy
and this isn't a one-way street. The ADP gets recharged.
constantly. It's the ATP-ADP cycle. The ADP molecule immediately gets sent back, mostly to the mitochondria, to have that third phosphate group stuck back on using energy from the breakdown of food. It's happening nonstop.
And the scale of this recycling is just mind-boggling. I remember reading this.
It really is staggering. Just to stay alive doing nothing special, the average human body makes and breaks down an amount of ATP roughly equal to its own body weight every single day.
Your body weight in ATP every day. That's incredible if that process stopped.
We'd be done in minutes. Seriously. It powers almost everything.
So what are some of the biggest energy hogs in the cell? Where does all this ATP actually go?
Well, a huge consumer is active transport, moving stuff across cell membranes against our natural flow.
Like a sodium-potassium pump.
Exactly like the sodium-potassium pump. That pump is running constantly in almost all our cells, especially nerve and muscle cells. It maintains the electrical potential across the membrane, which is absolutely fundamental for nerve impulses, muscle contraction, just basic cell stability. No ATP, no pump, nervous system shuts down.
Okay, what else? Building things.
Yep, all the synthesis, making proteins, replicating DNA, building complex molecules, that takes energy. Then there's actual mechanical work, like muscle fibers sliding past each other, or tiny motor proteins walking along tracks inside the cell carrying cargo.
And ATP is even involved in communication.
It is. It can be used as a signaling molecule itself, or it's the precursor for other important signaling molecules like cyclic AMP, cAMPA, and it's involved in neurotransmission too.
What's really neat is how the system regulates itself. It doesn't just run full blast until it crashes. High ATP levels actually put the brakes on production.
Yeah, it's a very elegant negative feedback loop. When ATP levels are high, meaning the cell's energy charge is good, the ATP molecule itself acts as what we call an allosteric inhibitor.
Okay, allosteric inhibitor. Break that down for us. What does it mean in practice?
It means the ATP molecule physically binds to certain key enzymes in the energy production pathway, but not at the enzyme's main working site, the active site. It binds somewhere else on the enzyme.
Like a side pocket.
Kinda, yeah.
Yeah.
And when it binds there, it changes the enzyme's shape slightly, sort of locking it in an off position. A key enzyme this happens to is phosphofructokinase 1, or PFK1, which is a major control point early in glycolysis.
So lots of ATP floating around basically tells PFK1, hey, take a break, we're good for now.
Exactly. It signaled the whole assembly line to slow down because the end product is abundant.
Right.
Then when the cell starts using up ATP quickly and levels drop, those ATP molecules detach from the enzyme, it snaps back into its active shape and production ramps right back up. It's self-correcting.
All right, so we know the currency, ATP, and the main bank where it's generated, the mitochondrion. Now let's walk through the actual assembly line step by step. The 4 stages of cellular respiration that turn food energy into ATP.
Okay, stage 1, glycolysis. This happens out in the main body of the cell, the cytoplasm. And it's ancient, evolutionarily speaking. It doesn't need oxygen, so it's anaerobic.
So this pathway was probably around long before Earth had much oxygen in its atmosphere.
Very likely, yes. It's the first sort of quick and dirty way to get some energy out of glucose. It involves a sequence of 10 specific enzyme-catalyzed reactions.
And the basic idea is just splitting the sugar molecule.
That's it. You take one molecule of glucose, which has 6 carbon atoms, and you split it into 2 smaller molecules, each with 3 carbons. These are called pyruvate.
But it's not free energy, right? You have to put some energy in first.
Correct. There's an initial energy investment phase. You actually spend 2 ATP molecules early on to kind of destabilize the glucose and get it ready for splitting.
Okay, spend 2 ATP to make. What?
Well, the second half, the energy payoff phase, generates 4 ATP molecules through a process called substrate-level phosphorylation. So the net gain from glycolysis itself is 2 ATP.
2 ATP doesn't sound like much compared to the total yield we hear about.
It isn't, but it's fast. And besides the 2-net ATP, glycolysis also produces two molecules of something called NADH, plus, of course, the two pyruvate molecules.
And those NADH molecules are important later, right? They're carrying energy.
They're carrying high-energy electrons that will be cashed in during the final stage for a much bigger ATP payoff. Think of NADH as energy checks written during glycolysis.
Okay, so glycolysis gave us 2 ATP, 2 NADH, and 2 pyruvate all out in the cytoplasm. To get the big energy prize, that pyruvate needs to go somewhere else.
Right. If oxygen is available, those two pyruvate molecules are actively transported from the cytoplasm
Mm.
across both mitochondrial membranes and into the very center of the mitochondrial matrix.
So inside the powerhouse now, what happens to pyruvate there?
It undergoes a crucial conversion step. It's often called pyruvate oxidation, or the link reaction. Each pyruvate molecule is converted into a two-carbon molecule called acetyl-CoA.
Pyruvate has 3 carbons, acetyl-CoA has 2. What happens to the third carbon?
It gets oxidized, combined with oxygen, and released as carbon dioxide, CoA. That's actually one of the sources of the CO we breathe out. And importantly, during this conversion, another molecule of NADH is generated for each pyruvate.
So this isn't a major ATP-producing stage itself, it's more of a prep step for the next big cycle.
Exactly. It's bridging glycolysis to the main event in the matrix. So starting from that one glucose molecule, this stage yields, remembering there were two pyruvates, 2 acetyl-CoA molecules, 2 molecules of CoA, and 2 more NADH. The acetyl-CoA is now carrying the remaining energy potential from the original glucose.
Okay, now we're really deep inside the mitochondrion, in the matrix, with these 2 acetyl-CoA molecules. They feed into the famous Krebs cycle, right? Also called the citric acid cycle.
That's the one. This cycle is often taught as, this complex diagram with lots of intermediates and enzymes, which can be intimidating.
Yeah, I remember trying to memorize all those steps, but what's the big picture? What's its main job?
Its main job is to finish oxidizing the fuel, and in doing so, harvest as many high-energy electrons as possible. It's really the cell's primary factory for electron carriers.
How does it work, basically?
It's a cycle, meaning it starts and ends with the same molecule. Each 2-carbon acetyl-CoA joins up with a 4-carbon molecule called oxaloacetate. That makes a 6-carbon molecule, citrate, hence the name citric acid cycle.
Okay.
Then through a series of 8 enzyme-driven steps, those 2 carbons from the acetyl group are systematically stripped off and released as co-urose. Along the way, the cycle regenerates the starting 4-carbon oxaloacetate molecule, so it's ready to accept another acetyl-CoA.
And during those steps, that's where the energy harvesting happens.
Yes. With each turn of the cycle, and remember it turns twice for every glucose molecule, once for each acetyl-CoA, it generates a little bit of direct energy currency, usually 1 molecule of GDP, which is easily converted to ATP. So that's 2 ATP or GDP per glucose.
This still sounds pretty low on the direct ATP count.
It is, because the real payoff of the Krebs cycle isn't the direct ATP, it's the massive Hall electron carriers. For each glucose molecule, meaning 2 turns of the cycle, the Krebs cycle churns out 6 NADH molecules and 2 molecules of another electron carrier called FADH.
Okay, so let's tally up. After glycolysis, pyruvate oxidation, and the Krebs cycle, we have a net of 4 ATP so far.
Right, 2 from glycolysis, 2 from Krebs.
But we've also accumulated a lot of these electron carriers, 2 NADH from glycolysis, 2 from pyruvate oxidation, and now 6 NADH and 2 FADH from the Krebs cycle.
Exactly. That's a total of 10 NADH and 2 FADH per glucose, all loaded up with high-energy electrons, ready for the final stage where the big money is made.
And this final stage, stage 4, is where the vast majority of ATP gets synthesized. This is oxidative phosphorylation. And it happens on that highly folded inner mitochondrial membrane, right? Those cristae.
Precisely. This is why those folds are so important. They provide the massive surface area needed for this stage. Oxidative phosphorylation actually consists of 2 tightly coupled processes.
Okay, what's the first part?
The first part is the electron transport chain, the ETC. All those NADH and FADH molecules we collected. They now deliver their high-energy electrons to a series of protein complexes embedded right in that inner membrane.
Like a bucket brigade passing electrons down a line.
Kinda, yeah. The electrons get passed from one complex to the next, moving to slightly lower energy levels each time. And the energy released during these handoffs isn't wasted.
What does the cell do with that released energy?
It uses it to actively pump protons, hydrogen ions, ATP from the mitochondrial matrix across the inner membrane into that inner membrane space.
So it's building up a high concentration of protons in that space between the inner and outer membranes.
Exactly. You get way more protons outside in the inner membrane space than inside in the matrix. This creates two things simultaneously: a concentration gradient, more protons outside, and an electrical gradient, more positive charge outside. Together, we call this the proton motive force.
Okay, proton motive force. That sounds like stored energy, like water building up behind a dam.
That's a perfect analogy. The inner membrane is the dam. The ETC protein complexes are the pumps pushing water, protons, uphill into the reservoir, the intermembrane space. And that reservoir, full of pressurized water, represents the potential energy of the proton motive force.
So the electrons provide the energy for the pumps. What happens to the electrons once they've gone down the whole chain and lost their energy?
They have to go somewhere, otherwise the whole chain gets backed up. At the very end of the ETC, the now low-energy electrons are passed to the final electron acceptor, which in us is molecular oxygen.
Ah, so that's why we need to breathe oxygen.
That is fundamentally why. The oxygen accepts the electrons, combines with some protons from the matrix, and forms water. Without oxygen, to take those electrons at the end, the whole electron transport chain grinds to a halt and ATP production crashes. That's why we call it aerobic respiration.
Okay, so the ETC built up this proton reservoir, this proton motive force. How does the cell cash that in for ATP?
That's the second part, called chemiosmosis. The protons naturally want to flow back down their concentration and electrical gradient from the intermembrane space back into the matrix. But the intermembrane dam is largely impermeable to them.
So there must be a specific gate or channel they can go through.
Exactly. There's a remarkable enzyme complex also embedded in the intermembrane called ATP synthase. This is the turbine in our dam analogy.
Right.
Protons can only flow back into the matrix by passing through ATP synthase. And as they flow through, they cause part of the enzyme to physically rotate, like water spinning a turbine.
And that rotation does the work.
Yes. That mechanical rotation drives the catalytic part of the enzyme, forcing ADP and inorganic phosphate together to synthesize ATP. It's an amazing conversion of electrochemical potential energy into mechanical energy and then into chemical energy stored in ATP.
And this stage makes a lot of ATP compared to the others?
Oh, absolutely. This oxidative phosphorylation process, driven by the proton flow through ATP synthase, generates the lion's share estimates are typically around 28 to 34 ATP molecules per original glucose molecule. That's why oxygen is so critical for sustained energy production.
Okay, that assembly line is incredible. But it needs fuel to run. Our bodies are amazing because they can use different types of fuel, carbs, fats, even proteins. Let's get into how each of these gets fed into the system and, importantly, why the body might prefer one over another at different times.
Right, let's start with carbohydrates, mainly glucose. This is generally the body's preferred quick access fuel source.
Why preferred?
Well, partly because it's versatile. As we saw, glucose can start being broken down anaerobically through glycolysis. That gives you a rapid, though small, energy burst, even if oxygen is limited for a moment. Fats and proteins need oxygen right from the start of their main breakdown pathways.
Okay. Now, about that ATP yield from glucose. We often see different numbers in textbooks or articles. Sometimes it's 38 ATP, sometimes 36, sometimes lower, like 30 or 32. What's the real story there?
Yeah, that variation causes a lot of confusion. The older, higher numbers, like 38, were often theoretical maximums based on simple calculations. More realistic modern estimates put the net yield closer to 30 to 32 ATP per glucose molecule for most cells.
And why the difference? Where does the energy cost come in?
A big part of it is the energy needed to transport things across the mitochondrial membranes. Remember those two NADH molecules produced during glycolysis out in the cytoplasm?
Yeah, they need to get into the matrix to drop off their electrons at the ETC.
Exactly. And getting them across the inner mitochondrial membrane isn't free. The cell uses special molecular shuttle systems to do this. There are two main ones: the malate aspartate shuttle, which is more efficient, and the glycerol 3-phosphate shuttle, which is a bit less efficient energetically.
So which shuttle system a particular cell uses affects the final ATP count?
Precisely. Different tissues might rely more on one shuttle than the other. So that transport cost accounts for a couple of ATP difference in the final tally. It's a real biological detail, not just different ways of counting. It reflects that slight trade-off between, say, speed and maximum efficiency in different cellular contexts.
Okay, so carbs are the quick, versatile fuel, but fats, lipids, they're the body's long-haul energy source, right? The big reserves.
Absolutely. Fats are our largest energy reserve by far and the most energy dense. They're the main fuel source when we're resting, sleeping, doing low-intensity activity, or if we're fasting for a while.
How does the body tap into this dense reserve? What's the process called?
It's called beta oxidation. First, the fatty acids, which are the main component of fats used for energy, have to be activated in the cytoplasm. This actually costs a little energy up front, the equivalent of about 2 ATP. Think of it as an investment to get the process going.
Okay, activation costs 2 ATP. Then what?
Then the activated fatty acid is transported into the mitochondrial matrix. And once inside, it enters what's called the beta oxidation spiral.
A spiral, not a straight line or a simple cycle.
Well, it's cyclical in a way. In each round of the spiral, enzymes chop off a 2-carbon unit from the end of the fatty acid chain. This 2-carbon unit is released as acetyl-CoA.
Ah, acetyl-CoA, the same molecule that came from pyruvate oxidation and feeds into the Krebs cycle.
Exactly. That's the beauty of it. It plugs right into the existing machinery. And each round of beta oxidation doesn't just produce acetyl-CoA. It also generates 1 molecule of NADH and 1 molecule of FADH.
So it's systematically breaking down the long fatty acid chain, 2 carbons at a time, and harvesting electron carriers along the way.
Precisely. It keeps spiraling, chopping off 2-carbon units until the entire fatty acid chain is converted into acetyl-CoA molecules, plus all the NADH and FADHs generated during the chopping process.
And then all those products just feed into the regular pathway.
Yep. The acetyl-CoA goes into the Krebs cycle, and the NADH and FADHs go straight to the electron transport chain. Because fatty acid chains are typically very long, often 16 or 18 carbons or more, you get a huge number of acetyl-CoA units and electron carriers from just one molecule.
Which translates to a massive ATP yield.
Massive is right. Take palmitic acid, for example, a common 16-carbon saturated fatty acid. Its complete oxidation yields somewhere around 106 net ATP molecules. Compare that to the 3032 from glucose. It really shows you the incredible energy density of fat.
Okay, carbs are fast, fats are dense. What about protein? Our bodies are built of protein, so using it for fuel seems counterproductive.
It generally is. Protein's main job is building and repairing tissues, making enzymes, hormones, all that structural and functional stuff. The body really only turns to protein for energy under duress, like prolonged starvation, when carb and fat stores are running very low, or maybe if someone eats an extremely high-protein diet with insufficient carbs or fats. It's the fuel of last resort.
Why last resort? Is it inefficient?
It's metabolically costly. The first unavoidable step is deamination. You have to remove the nitrogen-containing amino group from the amino acid because only the remaining carbon skeleton can be used for energy.
Okay, remove the nitrogen. What's the problem with that?
The problem is that the removed amino group becomes ammonia, NHAs, which is highly toxic, especially to the brain.
Ah, so the body has to deal with this toxic byproduct immediately.
Exactly. It can't just let ammonia build up. So the liver takes that ammonia and converts it into a much less toxic substance called urea through a process called the urea cycle.
And that cleanup process costs energy itself.
Yes, the urea cycle consumes ATP. So you're spending energy just to safely dispose of the nitrogen waste before you even get energy out of the carbon part. That's part of the cost.
Okay, so after deamination and dealing with the ammonia, what happens to the leftover carbon skeleton?
The remaining carbon structure can then enter the main energy pathways. Depending on which of the 20 amino acids it came from, its skeleton might be converted into pyruvate or acetyl-CoA, or it might enter the Krebs cycle directly as one of the cycle intermediates like alpha-ketoglutarate.
So it finds a way in, but it's a complicated and energetically expensive detour.
Very much so. It works when needed, but it's definitely not the body's preferred way to generate ATP due to the extra steps and the energy cost of managing the nitrogen waste.
Okay, let's zoom out and compare these 3 fuels head to head. We already established fat wins on energy density, right? More calories and thus more ATP per gram.
Correct. About 9 kilocalories per gram for fat versus only about 4 kilocalories per gram for carbohydrates and protein. So gram for gram, fat packs the most energy punch.
But you mentioned earlier there's another way to look at efficiency, and it's crucial. Something about oxygen.
Yes, this is the real aha moment, I think. While fat yields more ATP per gram, carbohydrates yield more ATP per molecule of oxygen consumed. They are more oxygen efficient.
More ATP per unit of oxygen. How does that work?
It comes down to the chemistry. Carbohydrates are already partially oxidized molecules compared to fats, which are highly reduced. Think of it like carbs are already partway burnt. So they require less oxygen to be fully oxidized to co-Euros and water for the amount of energy they release. Fats need significantly more oxygen to be fully broken down for the same amount of energy yield, though they yield more total energy overall.
Okay, I think I get it. But why does oxygen efficiency matter so much in practice? Don't usually have enough oxygen.
Usually at rest or during low-intensity activity, yes. But it becomes absolutely critical during high-intensity exercise. When you're sprinting or lifting heavy weights or doing anything near your maximum effort, your muscles are demanding ATP incredibly rapidly.
Faster than your heart and lungs can deliver oxygen.
Exactly. Oxygen delivery becomes the bottleneck, the limiting factor. In that situation, your muscles need to get the absolute biggest bang for their buck from every single oxygen molecule that arrives.
And because carbs are more oxygen efficient, they become the preferred fuel under those high-demand, oxygen-limited conditions.
Precisely. That's why glucose is indispensable for high-intensity bursts. The body dynamically shifts its fuel preference based not just on what's available, but also on the intensity of the activity and the availability of oxygen. Fats dominate during endurance, carbs take over during sprints. It's a constant, context-dependent balancing act.
Okay, we've got the fuels in the assembly line. But like any complex factory, this cellular energy production relies on more than just the big machines. It needs smaller tools, catalysts, helpers.
Absolutely. The enzymes that run all these reactions, glycolysis, Krebs cycle, etc., often need non-protein helpers called cofactors or coenzymes to function properly. And this is where essential micronutrients, the vitamins and minerals in our diet, become critically important.
They're like the essential cogs and lubricants for the metabolic machinery. Without them, the whole thing can grind to a halt, even if you have plenty of fuel. Hashtag 4.1, the B vitamin complex, coenzymes for catabolism.
And the B vitamins are probably the best example of this. They are water soluble, which means our bodies generally don't store them well, so we need a regular intake. Many of them are direct precursors to key coenzymes involved in energy metabolism.
Let's go through some key examples. How vital are they?
Let's start with thiamine, B1. Its active form, TPP, is an essential cofactor for a huge enzyme complex called the pyruvate dehydrogenase complex.
That's the one that links glycolysis to the Krebs cycle, converting pyruvate to acetyl-CoA, right? Stage 2.
Exactly. Without enough thiamine, B1, that enzyme complex doesn't work properly. Pyruvate builds up, it can't enter the Krebs cycle efficiently. So carbohydrate metabolism basically hits a massive roadblock right there.
Wow. Okay, what about the electron carriers, NADH and FADHs? They seemed pretty central.
They absolutely are. And they are completely dependent on B vitamins. Riboflavin B2 is needed to make FAD, the molecule that becomes the electron carrier FADHers when it accepts electrons, mostly in the Krebs cycle and beta oxidation.
And niacin.
Niacin B3 is required to synthesize NADO, which becomes the most abundant electron carrier, NADH, collecting electrons from glycolysis, pyruvate oxidation, and the Krebs cycle. If you're deficient in B2 or B3, you simply can't make enough of these electron carriers.
So it's like having plenty of fuel, glucose or fat, but not enough trucks, NADH and FADH, to transport the energy to the final power plant, the ETC.
That's a perfect analogy. You break down the fuel, but you can't capture and transport the released energy effectively. And then there's pantothenic acid, B5.
What does B5 do?
B5 is a central component of coenzyme A. CoA. Remember, acetyl-CoA, the CoA part comes from pantothenic acid. Coenzyme A acts like a handle, carrying those two carbon acetyl groups into the Krebs cycle. No B5, no CoA, no way to efficiently get fuel into the main aerobic engine.
And B6. You mentioned protein earlier.
Yes, pyridoxine B6 is involved in tons of reactions, over 100, but it's especially vital for amino acid metabolism. It helps with those transamination reactions needed to shuffle amino groups around and prepare the carbon skeletons of amino acids to enter the energy pathways if needed.
So taken together, a deficiency in B vitamins isn't subtle. It directly cripples energy production at multiple key points.
Absolutely. The symptoms make perfect sense biochemically. Fatigue, muscle weakness, poor concentration, neurological issues in severe cases. The cellular engines are sputtering.
Okay, B vitamins are crucial coenzymes. What about minerals? Are there key minerals acting as cogs in this machine?
Definitely. Two stand out for energy production: iron and magnesium. Let's take iron Phe first.
Yeah.
We often think of iron just for hemoglobin carrying oxygen in the blood.
Right, preventing anemia.
But iron is absolutely essential inside the cell for using that oxygen, specifically within the electron transport chain. Its magic lies in its ability to easily flip between two oxidation states, FeO and FeO. This allows it to readily accept and donate electrons.
So it's acting as a conductor within the ETC.
Exactly. Iron is found in two key places in the ETC complexes. First, as part of heme groups within proteins called cytochromes. Second, in structures called iron-sulfur, FeS clusters, found in complexes 1, 2, and 3. These iron centers are literally the stepping stones that electrons hop along down the chain, releasing energy.
So if someone is iron deficient, it's not just about getting oxygen to the tissues, it's also about the tissues being able to use the oxygen effectively in the ETC.
Precisely. Low iron compromises the very structure and function of the electron transport chain. The electrons can't flow smoothly.
Is there data on how much this impacts actual ATP production?
Yes, and it's quite dramatic. Studies, for example, on human heart muscle cells, which have incredibly high energy needs, showed that inducing iron deficiency led to a staggering 74% reduction in cellular ATP levels.
74%. That absolutely explains the profound fatigue of iron deficiency. It's a cellular energy crisis.
It really is. Now, the other key mineral is magnesium, MgRO. It's involved in, I think, over 300 different enzyme reactions in the body.
Wow. How does it specifically impact energy?
Its most fundamental role is actually with ATP itself. Inside the cell, ATP usually doesn't exist as a free molecule. It's almost always bound to a magnesium ion, forming the Mg-ATP complex.
An Mg-ATP, not just ATP.
Right, and for most enzymes that use ATP, especially the ones called kinases, which transfer phosphate groups, it's this Mg-ATP complex that is the actual biologically active molecule they recognize and work with.
That's a critical point. It means even if you make enough ATP, if you don't have enough magnesium, the cell can't properly use the ATP it made.
Exactly. The energy currency can't effectively plug into the machinery that needs it. On top of that, magnesium ions also act as essential cofactors for several key enzymes in the ATP production pathways themselves, like hexokinase and PFK1 back in glycolysis, and some Krebs cycle enzymes too.
So a magnesium deficiency hits energy from both ends, making it and using it.
Correct. It can hobble production at key control points and also impair the utilization of whatever ATP is produced. This contributes to symptoms like muscle weakness, cramping, fatigue, all signs of impaired cellular energy handling. Hashtag 4.3 coenzyme Q10, ubiquinone.
Okay, B vitamins, iron, magnesium. There's one more helper molecule often mentioned in the context of mitochondria, coenzyme Q10. Where does that fit in?
Coenzyme Q10, also known as ubiquinone, plays a really unique and essential role. It's a lipid-soluble molecule that lives within the inner mitochondrial membrane, and it acts as a mobile electron carrier within the electron transport chain.
Mobile, so it moves around, unlike the big protein complexes.
Yes. Its job is to shuttle electrons between the major complexes. Specifically, it picks up electrons from complex 1, which receive them from NADH, and also from complex 2, which receive them from FADH via fusinate.
Okay.
And then it physically moves through the membrane and delivers those electrons to complex 3. It's the crucial link connecting the first half of the chain to the second half.
So without CoQ10, electrons from both NADH and FADH row would hit a dead end after the first couple of steps.
Pretty much. The flow would be severely bottlenecked. Its mobility makes it perfect for collecting electrons from multiple entry points and funneling them forward.
Does it have any other roles?
It does have a very important secondary benefit. In its reduced form after it has accepted electrons, it's called ubiquinol CoQ10, acts as a powerful antioxidant right there in the mitochondrial membrane.
Protecting the energy factory from damage.
Exactly. The ETC, while essential, inevitably produces some reactive oxygen species, free radicals, as byproducts. Ubiquinol helps neutralize these damaging molecules right at the source, protecting the delicate mitochondrial membranes and proteins from oxidative damage. So it's vital for both efficiency and protection.
All right, this has been incredibly detailed. We've established the cellular engine mitochondria, ATP, the metabolic pathways, the cofactors. It seems like a pretty steady, continuous process, assuming you have fuel and the necessary helpers.
Right. The underlying machinery is designed for continuous, regulated operation.
So now we absolutely have to tackle that paradox we started with. If the engine is steady, why do we feel like we're on an energy roller coaster? Why the dramatic crashes and surges in subjective energy?
This is where we bridge from the cellular mechanics to the whole body system, especially the brain and hormones. The key insight is that your perceived energy state isn't just a direct readout of your total cellular ATP levels across the body.
It's more about fuel delivery to the brain and brain chemistry.
Exactly. It's largely dictated by rapid changes in fuel availability to the brain specifically, and by the state of your neurochemistry, things like motivation and alertness signals. These dramatic shifts are often top-down phenomena, orchestrated by the neuroendocrine system, hashtag tag 5.1, the neuroendocrine stress response. The rapid surge.
Okay, let's pick the rapid surge first. That feeling when you snap from feeling down or tired to suddenly feeling awake, focused, motivated, maybe because of an urgent deadline or something exciting happening. What's causing that biochemically?
That's often the work of the acute stress response system. It has two main branches. The first one is super fast, almost instant. The sympathetic adrenal medullary SAM system.
This is the fight or flight response.
That's the one. Your brain perceives a challenge or threat, signals the adrenal glands, and they dump a flood of adrenaline, epinephrine, and noradrenaline into your bloodstream.
We feel that as increased heart rate, maybe sweaty palms. But how does it mobilize energy?
Adrenaline is a powerful metabolic messenger. It hits the liver and muscles and tells them to rapidly break down stored glycogen, that's glycogenolysis. It also signals fat cells to release fatty acids into the blood.
So it instantly floods the system with readily usable fuel, glucose, and fats.
Instantly. It's like opening the floodgates on the body's quick-access energy stores, ensuring the brain and muscles have abundant fuel for immediate action. That's the biochemical basis of that sudden on feeling.
Okay, that's the immediate surge. Is there a follow-up system?
Yes. The second branch is a bit slower but more sustained. The hypothalamic pituitary adrenal HPA axis. This pathway leads to the release of cortisol, often called the stress hormone.
And what's cortisol's role in energy? Is it also about mobilizing fuel?
It is, but in a more sustained way. Cortisol helps keep blood sugar levels up over the medium term. It promotes gluconeogenesis, the liver making new glucose, often from amino acids. And it also helps the brain utilize that glucose effectively. So adrenaline gives the initial KCK, cortisol helps maintain the readiness, hashtag tag 5.2, the glucose-insulin axis, the crash.
Okay, that explains the surge. What about the opposite, the sudden crash? Feeling energetic one moment, then hitting a wall of fatigue an hour or two later, especially after eating certain foods.
Ah, yes, the infamous sugar crash. This is usually driven by the interplay between glucose and insulin, particularly after eating foods with a high glycemic index, GI.
These are foods that break down quickly into sugar, like white bread, sugary drinks, refined cereals.
Exactly. They cause a very rapid and high spike in your blood glucose levels.
Yeah.
Your body sees this sudden sugar flood as potentially dangerous, an emergency.
So it mounts a counterattack.
Precisely. The pancreas responds by releasing a large surge of the hormone insulin. Insulin's job is to get that excess glucose out of the bloodstream quickly, pushing it into cells like muscle and fat cells for storage or immediate use.
But sometimes the response is too strong.
That's the problem. Sometimes the insulin surge is so large or so rapid that it clears the glucose too effectively. It causes blood glucose levels to plummet, often dropping below the normal baseline level. This is called reactive hypoglycemia.
Reactive hypoglycemia. And the brain really doesn't like low blood sugar, does it?
It hates it. The brain relies almost exclusively on a steady supply of glucose for its fuel. When blood glucose drops suddenly, even if it's temporary, the brain interprets it as a major fuel shortage, basically starvation.
And the symptoms of that brain starvation are what we feel as the crash.
Exactly. That's where the profound fatigue, the lethargy, the irritability, what people call hanger, comes from. And also that feeling of brain fog, the inability to concentrate or think clearly. The brain's function is directly impaired by the lack of stable fuel.
And this whole cycle, the sugar spike, the big insulin surge, the blood sugar crash, it can happen relatively quickly, right?
Oh, yeah. Within maybe 90 minutes to 2 hours after eating that high GI meal, it perfectly explains those dramatic mid-morning or mid-afternoon energy slumps that so many people experience. Hashtag 5.3, the mood motivation matrix.
So we've got hormonal fuel mobilization explaining the surge and the glucose-insulin roller coaster explaining the crash. But there's another layer, isn't there? Our actual desire or motivation to use energy, even if it's available.
Absolutely. Our subjective sense of energy is deeply intertwined with our brain's motivational and mood circuits. You can have plenty of ATP circulating, but if your motivational neurochemistry isn't firing, you'll still feel flat and unenergetic.
And the key player in motivation is usually dopamine.
Dopamine is central. It's often called the motivation molecule. It's released, interestingly, not so much when we get a reward, but in anticipation of achieving a goal or receiving a reward. High dopamine levels are associated with drive, focus, goal-directed behavior, and a feeling of wanting to engage.
And low dopamine.
Low dopamine levels are linked to apathy, lack of motivation, difficulty initiating tasks, even if physically the energy is there.
What about other mood regulators like serotonin?
Serotonin plays a role too, more related to feelings of well-being, calmness, and emotional stability. The balance and interplay between neurotransmitters like dopamine and serotonin significantly color our perception of our energy levels and our willingness to exert effort.
Okay, so let's put it all together for someone experiencing that rapid shift from feeling low to feeling motivated and energetic. It probably wasn't a sudden miracle of ATP production in their cells.
Almost certainly not. That low feeling might have been a combination of factors, maybe slightly low blood sugar from a previous meal's insulin response, maybe low environmental stimulation leading to lower dopamine tone.
And then the shift happens.
The shift is often triggered by something psychological, a new, interesting task, an engaging conversation, a sudden deadline. This activates the brain's dopamine system. Okay,
Wow.
there's a potential reward here.
Which provides the motivation.
Right. And that mental activation itself can trigger a mild stress response, the same HPA systems we talked about, that mobilizes some stored glycogen and fat, ensuring the brain gets the fuel it needs to support that new, motivated state.
So the brain gets motivation and fuel delivered almost simultaneously, creating that instant feeling of being on.
Exactly. It's a top-down orchestrated event that changes the subjective experience dramatically, even if the total body ATP turnover rate hasn't fundamentally changed all that much in that short period.
This is fascinating because it really shifts the focus. If we want sustained, reliable energy, chasing those temporary surges like with caffeine or sugar seems like addressing the symptom, not the root cause.
That's exactly right. Those things can provide a temporary lift by manipulating those top-down systems, but they don't build a more resilient foundation. For long-term vitality, we need to focus on cultivating stability within the underlying physiological systems.
And that brings us to concepts like metabolic balance and adaptability, #6.1 metabolic homeostasis and flexibility.
Right, two key concepts here. First is homeostasis. This is the body's constant effort to maintain a stable internal environment. In terms of energy, it's about continuously matching energy production to energy demand, keeping blood sugar stable, managing hormone levels like insulin and glucagon. It uses sophisticated sensors like AMPK in cells to monitor energy status.
And that sugar crash we talked about is basically a failure of homeostasis. The system overreacts trying to restore balance.
Precisely. It's an overcorrection, a sign the regulatory system is maybe being pushed too hard or isn't functioning optimally. The second related concept, which is maybe even more important for long-term energy stability, is metabolic flexibility.
Metabolic flexibility, what does that mean exactly?
It's the ability of your body, and specifically your mitochondria, to efficiently switch between burning different fuel sources, primarily carbohydrates and fats, depending on what's available and what the body needs at that moment.
So someone who is metabolically flexible can easily burn the glucose from a meal when it's available.
Right. And then a few hours later, as blood glucose naturally drops, they can seamlessly switch over to mobilizing and burning stored body fat for energy without any drama.
That sounds like the ideal state, being able to use whatever fuel is appropriate.
It really is. This adaptability, this smooth fuel switching, is a cornerstone of metabolic health and is strongly linked to having stable, sustained energy levels throughout the day. Tag 6.2, the consequence of inflexibility.
So what happens when people lose this flexibility, maybe due to diet, lack of exercise, chronic stress?
That leads to metabolic inflexibility. This is actually a hallmark feature of conditions like insulin resistance, prediabetes, and type 2 diabetes. Essentially, the body loses its ability to effectively switch away from burning glucose and ramp up fat oxidation when it should, like between meals or during fasting.
So they get kind of stuck burning sugar.
To a large extent, yes. They become overly reliant on glucose. And because they can't easily tap into their abundant fat stores when blood glucose levels naturally dip.
They experience it as an energy crisis.
Exactly. Even though they might have tens of thousands of calories stored as body fat, their cells can't efficiently access it. So when blood glucose falls, the brain screams, fuel shortage. This triggers intense hunger, especially cravings for quick carbs, along with fatigue, irritability, and brain fog.
It's a vicious cycle. They feel bad, crave carbs, eat carbs, get a temporary fix, but reinforce the underlying inflexibility.
That's the cycle. They're constantly riding that glucose-insulin roller coaster because their system has lost the flexibility to smoothly transition to using its fat reserves. Hashtag check 6.3 actionable protocols for resilience.
Okay, so if the goal is to break that cycle and build metabolic flexibility and stable energy, we need practical strategies. It sounds like it comes down to managing the inputs and training the system. Let's talk about the key pillars: nutrition, exercise, and recovery.
Absolutely. They all work together synergistically. Let's start with nutrition, focusing on stabilizing that fuel input.
Avoiding those big glucose spikes seems key.
Definitely. The first step is shifting towards low glycemic index GI foods. These are foods that break down more slowly, releasing glucose into the bloodstream gradually. Think non-starchy vegetables, legumes, whole grains, nuts, seeds. This leads to a more moderate, manageable insulin response, preventing that reactive hypoglycemia crash.
And how you combine foods matters too, right? Not just
Mhm.
eating carbs alone.
Crucial. Always try to combine macronutrients. Eating carbohydrates alongside protein, healthy fats, and especially fiber dramatically slows down how quickly the stomach empties. This physically blunts the speed of glucose absorption and smooths out the blood sugar curve. A balanced meal is much better than just carbs alone.
What about when we eat? Does timing matter?
There's growing evidence for nutrient timing. It seems our bodies, guided by circadian rhythms, tend to be more insulin sensitive earlier in the day. So consuming a larger proportion of your daily calories, especially carbohydrates, earlier rather than later might help improve glucose handling for some people.
And of course, we can't forget those micronutrients we talked about earlier, the B vitamins, iron, magnesium.
Non-negotiable. Ensuring micronutrient sufficiency through a diet rich in diverse whole foods is fundamental. You need those catalysts and cofactors for the metabolic machinery to run smoothly. Deficiencies create bottlenecks, regardless of your macros. Think lots of colorful plants, nuts, seeds, quality protein sources.
Okay, pillar one, stabilizing nutrition. Pillar two has to be exercise. How does movement help build flexibility?
Exercise is incredibly powerful, acting in both the short and long term. Short term, muscle contraction itself can pull glucose out of the bloodstream without needing insulin. It uses different pathways, like GLUT4 translocation triggered by contraction.
So, a walk after a meal can directly help lower blood sugar.
Yes, it's an immediate benefit for glucose control. But the long-term effects are even more profound. Regular exercise, especially a mix of endurance and some higher intensity work, is the most potent stimulus we know of for mitochondrial biogenesis.
Meaning the body actually builds more mitochondria?
Yes, and often better functioning mitochondria. Exercise triggers signaling pathways in the muscle cells, involving molecules like PGC1 that tell the cell, hey, we need more energy capacity. So the cell builds more powerhouses.
More mitochondria means better aerobic capacity.
And critically, it significantly enhances metabolic flexibility. Exercise trains the muscles to become much better at oxidizing fatty acids for fuel. So when glucose isn't readily available, the muscles can more easily tap into fat stores, reducing that dependence on carbs and promoting smoother fuel switching.
Okay, nutrition and exercise. The final pillar, often neglected, is recovery, particularly sleep. How does sleep tie into cellular energy and flexibility?
Sleep is absolutely not passive downtime. It's a period of intense cellular repair and waste clearance. During deep sleep, the body prioritizes processes like protein synthesis, tissue repair, and DNA damage control. And crucially, the brain activates its unique lymphatic system.
A lymphatic system, what's that?
Think of it as the brain's nighttime cleaning crew. It uses the flow of cerebrospinal fluid to flush out metabolic waste products and potentially neurotoxic proteins that accumulate between brain cells during the day. This is vital for maintaining healthy neuronal function.
And does sleep directly restore energy reserves too?
Yes, particularly for the brain. Sleep is essential for replenishing the brain's own stores of glycogen, which get used up during waking thought processes.
So skimping on sleep doesn't just make you feel tired, it actively impairs these vital repair, cleanup, and refueling processes.
Exactly. Chronic sleep deprivation messes up everything. It impairs cellular repair, allows waste products to build up in the brain, dysregulates hormones like cortisol and insulin, making metabolic inflexibility worse, and directly impacts cognitive function. It manifests as profound mental and physical fatigue, brain fog, and poor motivation, hitting all the factors we've discussed. Hashtag outro.
So, wrapping this all up, it seems the journey from feeling exhausted to feeling energized isn't usually about flipping a switch inside our mitochondria. That engine is generally running.
Right. The real story, the reason for those dramatic subjective shifts, lies higher up in the control systems. It's the brain, through the neuroendocrine system, and its sensitivity to the glucose-insulin axis that orchestrates the mobilization and delivery of fuel to meet perceived needs and governs our motivation to use it.
The mitochondrial ATP production is the foundation, but the brain and hormones are conducting the orchestra of our perceived energy state.
Beautifully put. And the path to sustained, reliable vitality isn't about constantly trying to manipulate that orchestra for a temporary high chasing the next sugar rush or caffeine jolt.
It's about building a stronger foundation.
Exactly.
Yeah.
It's about cultivating that deep metabolic flexibility and homeostasis, building a system that can smoothly switch fuels, that doesn't panic when blood sugar dips slightly, that has all the necessary micronutrient cofactors, and that gives adequate time for repair and regeneration during sleep. That's how you get off the roller coaster.
So the final thought for everyone listening, your body is always trying to find balance. If you feel like you're constantly fighting for energy, maybe think about which of those pillars, your nutrition patterns, your exercise habits, or your sleep quality might be the biggest bottleneck right now. Where's the main point of friction preventing your system from achieving that flexible, stable state?
Yeah, identifying that bottleneck is probably the most powerful first step you can take towards truly mastering your own energy.